Now I'm going to show you how you can use this in biology for a photosynthesis lab. This one could also be useful if you were studying geology or agriculture. So what I had done earlier was I took some spinach leaves and some red lettuce leaves, and I had soaked them in ethanol, just kind of grind them up, and then that produced cuvettes full of these wonderful colors. And then we can analyze these to see at which part of the spectrum they absorb the most light. There's a third cuvette here that's just filled with ethanol, and that's going to be our calibrator. So with the cuvette, I'm just going to slide that in smooth side towards the spectrum and then calibrate dark. You can see on the screen, just takes a few seconds. So that turns off the lights in the spectrometer, and then calibrate light. And basically what this does is it removes the ethanol from the spectrum in these two cuvettes. So now I'm going to pop that one out. And let's start first with the spinach, and let's look at the absorbance. So I have that here. If I wanted to switch that, I could just click there and that would switch it. So now I'm just going to, got the cuvette in there. I'm just going to hit record. And you can see really quickly that there's a lot of absorbance of blue light, and again, another peak here on the red end of the spectrum. I'm sure the biology teachers are not surprised at all, but this might be surprising to your students. So just hit stop on that one and label this one as spinach. And let's pull that out, see how simple this is. And then we'll put in the red lettuce leaves. And then again here, just hit record. And here we're getting a lot less absorbance from the red lettuce leaves. We can zoom in, can adjust the scale as needed. So you can see a lot of absorption on this end of the spectrum, and there's a little bit here, but you can see generally it has a different shape. If you wanted to compare, you can just stop recording and then click this button here, which show comparison mode. And then you can compare the absorption of light with the spinach, which is the top one, and the red lettuce, which is the bottom one. So you can think of some applications here in biology, in an agricultural course, or even in a geology course where you're going through the geological time scale, and why might we experience here leaves that absorb a lot of blue and red light, but not a lot of the middle of the visible spectrum where the sun is emitting a lot of its light. So that would lead into some great conversations with your students.